So, very warm welcome also from us to the Capital Markets Day. I'm really happy that I can be here and that our little Emma came on time. So, my name is Alexander Pelzer. I'm the head of R&D based here in Zwingenberg, and together with my colleague Ellis, VP Fermentation Technology based in Cardiff, we are delighted to give you insights in our unique technology and production platform. Erin has already introduced you to our unique customer offering. So we are offering 3 distinct yet interconnected services. And these include contract research, contract manufacturing, and products. And in each of these areas, we have absolute experts specialized in the respective field. And what's really important for us is that they work closely together to maximize synergies. And all of these services, CRO, CMO, and products, can rely on a shared technology platform. And we want to give you insights today into this shared technology platform, which is the engine of our businesses. I would like to start to introduce you to our advanced research and development platform, where we offer tailor-made solutions from discovery to production. We use these technologies for our contract research services, but of course also we use our R&D power to expand our product portfolio and develop innovative products. In end of 22, beginning of 23, we refined and sharpened our focus areas. And since then, we have been massively investing in them to develop new technologies. So we are focusing on enzymes and proteins, microorganisms and production strains, as well as bioprocesses and scale-up. And we have different teams specialized in different areas, as you can see here. So the team of enzyme technology works in enzyme discovery and enzyme engineering. The microbial strain development team works, for example, in bioprospecting, which is a technique to, for example, discover new microorganisms in nature. And this is a technology we have been using to develop the bioarchive, the huge strain collection we have. And how powerful microorganisms can be will be later on presented by Esther Gabor on the topic fermented food. We do strain engineering to specifically optimize strains for application, and one of our focus areas is production strain development, especially for enzymes, but also for other biomolecules. The bioprocess development team works on fermentation and downstream process development, and this also includes tech transfer and scale-up, especially to our production site in Cardiff. And everything we do is heavily supported by bioinformatics, which gets more and more relevant, a very broad and flexible instrumental analytics, quality management, and intellectual property management. And this is our advanced technology portfolio that is designed to deliver biosolutions. Then the question, of course, is where do we find these biosolutions? In nature, of course. So since day one, it has been, and it still is, our inspiration is the creativity and diversity of nature. So nature had over 3.5 billion time to solve numerous complex chemical problems, and we want to get access to nature's solutions, these biosolutions, and apply these solutions to our challenges, industrial challenges of today. This is the look back into evolution. What we see is that the future is digital. So we want to combine nature as a role model with the modern technologies of today. And one technology I would like to introduce you to is our MedExtra technology platform, because this platform is exactly doing this. MedExtra is composed of 3 modules: the database, MedExtra Design, and MedExtra Discover. The MedExtra database is digitized biodiversity that is continuously evolving. Here you can see some pictures of habitats that we have sampled, digitized, and stored on our servers here in BRAIN. And this is not only a database of a couple of hundreds of millions of sequences. The unique feature of our database is that it is very diverse. So as you can see here, 99.8% of the protein sequences that we have here in our database are not known in public domain. So if you want to get access to these novel sequences, you need to work with BRAIN. And this is what is for our customers very interesting, and also for us, of course. MedExtra Design is a toolbox of AI-generated enzymes. So these tools can be used to predict synthetic enzymes. And the beauty here is because we have so much novel sequence information no one else has, we can use this data to train these AI tools to come up with even better or other enzyme sequences no one else can provide. The MedExtra Discover module is in the end the filtering and selection module that helps us to find the right enzymes from database and design. So here you can see this is a bioinformatics workflow, how this normally looks like. We start with roughly 100,000 sequences, which is a lot, but then we need to filter out the right molecules and therefore use this bioinformatics pipeline. Normally for a typical project, we end up with 20 to 200 enzyme sequences that we want to work with. But we don't stop here. So we are not only a bioinformatics company, we then test it in real life. We bring it to the lab, express the genes, produce the enzymes, and characterize them in detail for application. So this is in a nutshell our advanced technology for discovering novel natural enzymes or novel synthetic enzymes for application. And we have shown that MedExtra works several times. One of the most impressive case studies, I would say, is the discovery of our own CRISPR-Cas-like genome editing tools, because this shows that it is really possible to develop truly new enzymes with MedExtra. We did exactly what I illustrated on the last slide. We went out in nature, sampled biodiversity, digitized it, put it into our MedExtra database, and used the Discover approach to identify over 2,000 CRISPR-Cas-like molecules. In the end, we focused on roughly 15 of them to develop them into genome editing tools that we can use here in BRAIN. And CRISPR-Cas9 got a Nobel Prize in 2020, is one of the disruptive technologies for medicine, plant and animal breeding, and also for industrial biotechnology, but the commercial licenses are so expensive. Therefore, we went through this workflow to finally got our own BMCs, brain metagenomic Cas, that we can use for production strain development. And as you can see here, we have developed then to work in our industrial enzyme production strains, but also in our other strains that we use for biomolecule production. In the end, the genome editing tool BMC enables precise and fast production strain development, and the biggest benefit for us is it significantly reduces production development times for our production strains. Not always are enzymes derived from nature perfectly fit for application. Therefore, we need sometimes protein engineering to specifically optimize them. And it is good to have both technologies at hand. Here you can see again this wheel of bioinformatics methods, because also for protein engineering, we use bioinformatics. We use something like multiple sequence alignments to analyze conservation and which amino acids in an enzyme we want to change and optimize enzyme characteristics. We use structure modeling to look at the structure of enzymes, the surface of enzymes, to predict characteristics. We also do something like docking studies, as you can see here, to predict which substrates can be converted into novel products. We use molecular dynamics simulations to analyze how flexible an enzyme is under certain conditions. And also here we use AI tools to predict which amino acids in an enzyme we need to tackle to optimize these enzymes. And in the end, the result of the workflow is the same. So the goal is to reduce the endless possibilities to what is beneficial for the enzyme, what is beneficial for the project. And for enzyme engineering, for example, we normally end up with 20 enzyme candidates that we then test in the lab. And here you can see an example for an alpha amylase for starch processing. As you can see here, the enzyme is quite huge, but due to the bioinformatics, we know only to focus on the red ones and the purple ones, because these are the positions we need to optimize to make the enzyme better for application. So to reliably provide our customers with novel enzymes or better enzymes, we have integrated both technologies, because enzyme discovery and engineering are equally powerful tools for providing novel enzymes. And of course, having both available greatly increases the chances of success. And this is possible that we can do discovery and engineering because these rely on the same technologies in the middle. And with this integrated strategy, we fuel enzyme innovation and produce market-leading enzyme solutions. As Arian already mentioned, creating or finding the best enzyme is only the first step. It even the most exceptional enzymes must be producible at scale. Otherwise, it's of little value for us. Therefore, these technologies are great, but you also need powerful industrial production strains and the bioprocesses to come to an enzyme production in the end. Therefore, we look at the whole pipeline: microbial strains, strain development, process development, manufacturing, and also product manufacturing. So at BRAIN Biocatalysts, the enzyme development is considered in a comprehensive and end-to-end manner. Let's have a quick look at our industrial enzyme production strains. We constructed in the last years so-called plug-and-produce systems. These are based on our industrial production hosts, standardized methods to come to a yes-no answer if an enzyme is producible or not in very short time frames. And our industrial production hosts, you can see right here, we use bacterial strains, yeast, and also fungi for enzyme production. To specifically optimize these strains, we use methods from expression technology, random mutagenesis, and also genetic engineering. The introduced BMC technology, the genome editing technology, plays a critical role here. So this is the enabler in genetic engineering. Looking at our bioprocess development platform, we normally start in very small volumes in the lab and do bioprocess development up to 200 liter scale. We have DSP development and also product purification development, full analytical support. I already mentioned the broad and flexible instrumental analytics that we have. And what we have, and this is really key for us, is experience with a wide range of production strains. As you can see here, we are not focusing only on one strain or two strains. For our industrial production host, we have several strains and a very experienced team who knows how to work with these strains. And what our customers really like is that we have extensive knowledge of industrial regulatory and quality requirements as well, because we know early on which media components to use, antibiotics to use, and things like this. And also here, like for the enzyme discovery and engineering, this is a fully integrated process, as you can see here. So during strain development, we directly also start the bioprocess development because this goes hand in hand. And to also give you a case study on this, here you can see only this middle part. So we have already identified a nice protein or nice enzyme. And then we go to our microbial plug-and-produce strains and check what is the initial situation. Is an enzyme producible, yes or no? And here you can see these black bands that we are able to produce low single-digit grams per liter of this protein already. So this is quite a good starting situation. And then over different rounds of strain engineering and process development, we end up with this very thick bands. And this is what industry is looking for. And this now is high double-digit grams per liter. I cannot really tell you the exact number because this is confidential. But as you can see here, this is a pretty good result in short time frames. With this, I wanted to show you that the integrated platform for production strains and bioprocesses is able to lead to high enzyme yields. A summary of our highlights. What we offer is a MedExtra technology platform, which is capable of identifying unique and high-performance enzymes for industrial application. We use an integrated enzyme discovery and engineering approach to drive success in delivering novel enzymes. We provide high-yield industrial production strains. We have our unique CRISPR-Cas tools here at hand for strain development, the BRAIN metagenomic Cas. And we use an integrated approach for production strain and bioprocess development that drives success in industrial enzyme production. And all of this is also offered to customers. And what makes this offer special is the range of technologies, the modularity, and the flexibility. We offer our customers custom tailor-made solution. This means we look at the challenge of the customer first. We can use our technology portfolio and help our customers to find the right combination of modules to tackle their challenge. And the beauty is that customers can select several modules to also go for the end-to-end solution from start to finish, but sometimes a solution can be quick and easy. So therefore, it is also possible to only select one of these modules. And one last comment on this. These technologies, our technologies, can be used across various industries. We are focusing on food and specialty enzymes, as Arian has already introduced you to, but we use these technologies also for pharma, chemical industry, food and feed, just to name a few. And with this, I would like to hand over to Andy Ellis, who's going to show you how this seamlessly connects to our fermentation manufacturing platform. Andy? Thanks.
Thank you very much, Alex. Given I think you had only 2 hours sleep last night? 3 hours sleep, oh wow. I thought you did exceptionally well there. So yeah, as Alex described, I want to give some insights into how the variety of R&D capabilities that have been discussed over the last few slides are fully integrated into the manufacturing capabilities for enzyme production. In terms of our enzyme manufacturing capabilities, we've invested heavily over the last 5 years, both in stainless steel, but also in our expertise, bringing in new talent and developing that talent to support the manufacturing side of the business. So Alex described the bioprocess optimization. Essentially, that happens within laboratory scale with very small fermenters in order to maximize the amount of value that we can get out of a particular production process. But of course, then we have to scale that up. And within Cardiff, we have a number of different pilot scale fermenters up to the 750 litre scale. And in reality, they are used for scale up, but actually in their own right can be used for manufacture. We have some incredibly high value, lower volume enzymes that we offer, particularly into the life science space, and those smaller fermenters, ideal scale for those particular products. Additionally, we have the 2 fully functional 10 meter cube fermenters, the nice shiny fermenters, I think as Arian described them, that you can see top center here. And additionally, a network of fermentation partners. We do have a number of enzymes that exceed our capacity. And over the last 12, going on for 15 years actually now, we've developed some really good partnerships to be able to push out our own strains and our own manufacturing processes to those manufacturers when we need that capacity. All of the fermentation capability at every single scale is supported by downstream processing technology. So what is downstream processing technology? Essentially, in the fermenter, you produce a relatively crude enzyme. And that needs recovering from the fermentation broth, but also needs purifying. And different applications, so delivering the science towards the applications, different types of applications require different levels of purity of the enzyme. So we have a different array of downstream processing assets and knowledge that allow us to fine-tune the purification specifically towards the application, the market, and the individual customer need. Over the last 18 months, we've specifically focused on a relatively new technology to the Cardiff site, which is chromatography. So chromatography is a particular type of purification, so downstream processing technology that allows for very, very high purity specifications to be achieved. And you can see an image there of the chromatography setup on the bottom left of the screen. All of these capabilities are compatible with the multiple production hosts that have been described by Alex. Behind the scenes, what we also have is a long history of expertise in product stabilization. Our product business is global, so we have to ship product, sometimes very high value product, overseas, and that product needs to be stable. Equally, when it's received by the customer, it needs to have the required shelf life stability. That's not always straightforward to be able to offer enzymes in such a stable fashion to meet those transport and shelf life requirements. But we have lots of knowledge in that space. Whether the enzyme is in liquid form or in powdered form, we have insights on how to get the best out of stabilizing them in their finished form. And to that end, again, over the last 18 months, We have fully commissioned and are fully exploiting our new freeze drying capabilities that can be seen at the top right of this particular slide. And as I mentioned earlier, all of this fully exploited by the pool of talent that we have across BRAIN Biocatalysts. I wanted to make a key feature of this point, design for manufacture framework. We feel it's an extremely important part of the biocatalyst offering. But what does it actually mean? Approximately 10 years ago, we hired a senior manager who come from the automotive industry. He had a background in engineering kind of design and development. And he embraced through his background and then brought it into the BRAIN Biocatalysts offering this framework that allows us to ensure we make the right informed decisions from the very start of any enzyme development project, right through strain development, through scale-up, ultimately towards manufacturing, to maximize the probability of success. What does this framework contain? Essentially, it's a set of decision-making criteria that are fully documented in work instructions and SOPs and guidance notes that ensure we capture the key parameters for success. So process economics, understanding the price point that a particular new enzyme which is required for the particular application. From that price point, maximizing the margins that we can deliver from the manufacture of that product, and working with the tools described by Alex around strain development and process optimization to hit both our internal margin targets and the customer target. So process economics, the whole design starts with understanding that framework. Similarly, in terms of environmental factors, both for the enzyme itself, the enzyme production process, and ultimately the application, we have to consider a number of environmental factors. And again, those play into the design rules. We operate in a heavily regulated environment. You know, we offer enzymes into the life science world and also into the food industry, and operate globally, as mentioned. So we have to ensure that we are compliant with the different geographical regulations, but also the particular market regulations that are applicable to each enzyme that we develop. And of course, intellectual property is also key. You know, Aryan mentioned our approach to kind of novel enzyme development, the funnel, the pipeline of NPD that was described, and mentioned that, you know, often we will look at a competitive enzyme and try to produce an alternate version of that with perhaps a special feature, but making sure that we don't step on any patent issues is really, really important. So ensuring FTO at the very start of the enzyme development process, and ideally developing some in-house know-how that we can protect is a key consideration. Quality framework, ensuring that, again, we design into the process a framework that ensures that the process itself operates under the required quality criteria, but also that the finished product hits the required quality specifications for its intended use. And all of that governed by time. Not every business that we come across as competitors, particularly the kind of smaller CRO and CMO business, has this kind of approach. In fact, we win some business whereby customers have gone to supplier A in the CRO and CMO space, and let's say misdesigned or not designed effectively for manufacture a particular enzyme, and we end up reverse engineering that. So we get a few projects based on our ability to approach enzyme development this way relative to some of the competition who don't. Another focal area for the fermentation side of the business, of course, is maximizing margins. This operates at two different levels in terms of capacity utilization and productivity. In terms of capacity utilization, we now have the two fully functional 10 meter cube fermenters. And since the second fermenter was fully commissioned just over 6 months ago, we put a lot of effort into debottlenecking the co-running of the two fermenters So maximizing the overlap of those two fermenters to ultimately increase capacity. And we've made some major breakthroughs there, particularly over the last 3 months. So a key focus area is maximizing the use of that capacity in Cardiff. But additionally, as mentioned several times, we utilize external capacity when we need to. And through our kind of planning tools, operational planning tools, we have kind of a push and pull strategy. And we take a holistic view on deciding strategically which processes to run externally versus internally, and often pull back processes from our external partner into the Cardiff capacity, if that makes sense to do so from a kind of holistic planning and from a margin perspective. So that's the capacity level. In terms of productivity, as mentioned previously through our approach for design for manufacture, we try to maximize fermentation margins from the get-go through the design of each individual enzyme manufacturing process, but it doesn't stop there. We're then committed to continuous improvement, to continue to drive margin improvement for the fermentation side of the business. And that operates at kind of 3 sub-levels. So strain development, you heard from Alex, how we can engineer the strain to make them more efficient. So that's a key activity that takes place across the BRAIN Biocatalysts team. Continuous fermentation optimization, so irrespective of the strain, tweaking the fermentation parameters to increase space-time yields, so maximizing the amount of value that we can obtain per meter cubed of fermenter per time is a really important consideration. The downstream processing, so the recovery and purification operations that come after the fermentation, also a key target area for improving margins. So we have cross-functional teams look at CI from engineers, planners, scientists working collectively to continue to drive the efficiency of the fermentation side of the business. And Ari and I had a discussion several weeks ago where he wanted a clear update on the goal that he'd set me at the start of 23-24, which was for a particular process targeting a significant productivity improvement on that process. And thankfully, the news was good. And I was able to disclose that we'd reduced the overall material costs by that process by 10% and a greater than 25% increase in overall process productivity. So we are getting good mobility on overall EBITDA improvement from the fermentation side. This will continue to be a key focal area for this financial year and beyond. So just to summarize some of those key points associated with our fermentation capabilities, we have a breadth of different fermentation scales towards food, enzyme needs, food specialties, and the life science industry. We have extensive downstream processing capability that can be used flexibly and dynamically depending on the end application requirements. This design for manufacture framework we're really proud of. We think it makes a difference. And a key focus on margin for the future, driving EBITDA growth from the fermentation side of the business. So just to kind of sum up how we kind of monetize all of the capabilities that we've described between Alex and I. Alex mentioned in his final slide the CRO business, so essentially where the R&D part of the platform can be monetized through one-to-one projects with clients, whereby the client is interested in the Zwingenberg team primarily, developing some innovative, novel intellectual property that is financed through project revenues and ideally ends up with a license deal. So that's a key model for monetizing those R&D capabilities. We have the CMO business, so the third kind of business model, which over the last 6 years has continued to go from kind of strength to strength in terms of interest and absolutely uses all of the manufacturing capability that we've described. And what does that look like? Well, essentially a customer will approach us. Typically they've already found an enzyme that they know kind of works, delivers for their application, but they don't know how to produce it. They need a partner to take that up to the relevant scale to access on a one-to-one exclusive basis commercial quantities of that enzyme. That's where BRAIN Biocatalysts fit in with the CMO business. Very close to Alex and I's kind of hearts in terms of the kind of passionate connection with the BRAIN Biocatalysts offering is the product side of the business. This is where we've spent, Alex, with our colleague Ludwig, a lot of time over the last 2 years is rejuvenating our product development pipeline for those go-to-market enzymes that Aaron referenced in his funnel diagram. We've got a real exciting bunch of enzymes in that enzyme development pipeline, and we're kind of looking forward to future kind of realization of those through go-to-market launches. Just wanted to close now on a couple of kind of case studies, give you some kind of real life examples of how we monetize all of what we've described. In this first one, it utilized both the R&D platform and the manufacturing platform. We engaged with a particular customer through CRO project mode and CMO project mode. Specifically, the customer requested that BRAIN Biocatalysts developed a novel enzyme for production of a particular food ingredient that will be used in gastrointestinal health. So the customer, one of the top 10 food and food ingredient businesses, they were particularly interested in ensuring that they had a patent or an IP position that they could lock down, ideally in a patented new enzyme technology and application thereof. So that was very much central to our design for manufacture principles when embarking upon this particular project. And we found the enzyme of interest through the MetExtra program. We, I think, screened 15 to 20 enzyme candidates, of which one of them hit all of the technical features that the customer required. We then developed the production strain. It was a bacterial production strain to effectively produce that enzyme for that particular customer, for that particular application. We scaled that up with bioprocess optimization towards the fermentation and the DSP. We produced several batches at the 10 meter cube scale of that enzyme for the customer's approval. And we're now, we were talking about this earlier, with the contracts department here, in negotiation terms for exclusive rights to that enzyme with that customer and ongoing supply contracts associated with that. It's a really good success story that covers many of the different facets that Alex and I have described. This is slightly different. Here, we identified a gap in the market for a particular enzyme for accessing a particular lipid substrate. So modifying lipid A to produce lipid B being very high value and having a certain application in the food industry. So we didn't work with any particular customer. This was in-house finance, so a kind of example of one of the funnel NPD enzymes mentioned by Aaron. Very specific quality criteria that we needed to take into account from the very start of the project. So as part of the DFM approach, quality stood out as being the main attribute. We developed the enzyme, developed the yeast strain for production of the enzyme, optimized, scaled up to the 10 meter cube scale internally. But what we also did is scaled up externally, because we had in mind this need to be flexible around where we actually produced that enzyme. So we scaled up to 20 meter cube scale, and then used the various production batches or trial batches from the different sites to put forward a regulatory submission that was granted, and we launched that product approximately 18 to 20 months ago, and we're now selling that to multiple customers in a number of different countries. And actually, Darren will talk, I think, more about the impact of this particular project in his slides later. So that brings, I think, a conclusion to our part of the presentation. Hopefully, we've given you a good insight into the different capabilities that we have on the R&D side and on the manufacturing side, and how those are fully integrated to serve the three different business models of CRO, CMO, and products. There's real strength in the technology platform. It's broad, it's deep, and we think it's a really high-performing engine that supports the BRAIN Biocatalysts business. Thank you for your time. Take any questions. Looks like we've baffled everybody. There's no questions online.
Thanks for the presentation and congrats on the happy event. So Nicolas Pauillac from Kepler Cheuvreux. Maybe a question, as you did a great job at teasing this very good platform that you developed. How does it work internally when you think about you have all of this possible enzyme? What's the decision process to say, okay, this one looks interesting, and how do you do, let's say, the arbitration between all of the possibilities? Is it more financial upside that you see, or it's also likelihood of it being realizable? Sorry.
Yeah, thanks for the question. So as you have seen, we have hundreds of thousands of opportunities where to start. So first step is always using our bioinformatics predictions to narrow it down to the relevant set of candidates. Important for us to mention here is that we use also a lot of AI-based tools, but we combine this with our experts' expertise, because what we still see, this huge AI trend is great for us. We need to follow it. We want to use it, but we always complement it with our expertise that our real humans have to say, okay, I think this enzyme has the property I'm looking for, or maybe this enzyme has here. So it's a combination of machines and humans, a lot of experience, to select a set of candidates. And then the next question is, how many enzymes can actually be screened? So for example, using a bacterial host, you can screen hundreds of enzymes. A fungal host like Aspergillus grows very slowly, then you cannot test so many enzymes because the project would take forever. Not forever, but pretty long times. And then this is also part of the decision process. In the end, of course, we also have to look at the commercial aspects later on. So how much enzyme secretion or production level needs to be reached that it makes commercially sense in the end. So there are many factors, starting from the database, bioinformatics, narrow it down, also use our expertise to predict which enzymes might do the job, and then looking at the application, what can be the strains or production strains we can use, looking at commercial parameters, which strains can actually be used, and what is required to come to the market in the end. So it's quite a complex process, and each step has opportunities to further narrow it down.
Maybe just to follow up. So it's more gaining efficiency and capitalizing on experience that you have rather than a major breakthrough, let's say.
It's both, but efficiency definitely, to screen as much enzymes as possible. So our strategy always is to predict as good as possible and screen as much as possible. That's our mantra.
Great. Thank you. I have two questions. The first one is on your CRO business. Could you share your view of the current market dynamics? We continue to hear from some larger players that they continue to see significant external headwinds, such as slower biotech funding and competitive pricing. So I'm wondering if you see something similar.
Yeah. So it is a more difficult market situation at the moment, I would say, because companies willing to fund contract research is challenging at the moment, but we see that there's a lot of interest in our technologies, which is fine, especially looking at our expertise in expression strains and production strains, enzyme production strains. So this is something where we see a clear market trend. There's a lot of competition in the field of enzyme discovery at the moment, especially because of the AI and data-driven technologies that are out there. But I think this is good because there are many companies also synergistically pushing this field together into the markets. And the good thing is that biodiversity is so diverse and so much enzymes are still there to be discovered that there is a lot of competition. But as you have seen in our database, 99.8% of the sequences are novel. So still, even though there are several companies working in this field, there's so much to discover still. So this is some market trends that I see at the moment. A lot of competition in the AI field, but this is fine because there's enough space for everyone. And very interesting is our technology at the moment around production strain development together with the scale-up opportunities that we can provide.
Yeah,
okay, great.
If I could just add to that point, or support that point, for me, the strain development that is now enabled by the guys in Zwingenberg in Alex's team is so powerful. I mean, for the product side of the business, you know, we now have access to an array of strains that we've never had before. And for our products customers, we're finding a lot of interest in this. So customers, particularly in the top segments or the larger food companies who've got interest in lots of different areas. We've been selling enzymes to those kind of companies for decades. And now we're able to talk about the strains for production of interesting molecules and food proteins. And that's creating quite a wave of interest, right? So the strains for me is a definite kind of unique offering that perhaps some of the other incumbents in the CRO space have not got that same level of strength, in my opinion.
Very clear, thanks. And my second question is on your product business, specifically on the second fermenter. Could you share your view on the utilization trajectory going forward?
Yeah, of course. It's a good question. We are pretty much at capacity with the first 10 meter cube fermenter. We are not quite at capacity with the second yet, so there's still capacity to fill. And we expect within the next 18 months to 48 months, we will fill that capacity. But of course, that kind of capacity is a kind of continuum. As I've kind of alluded to, we are continuously looking at process optimization, strain optimization, and so through relatively kind of low-cost investments, i.e. not in CapEx, we're able to expand that capacity. But that's the kind of timeline we're looking at for filling.
Thank you. 2 to 3 questions. Do you also use, for example, steel-sensitive microorganisms for production? Steel-sensitive organisms for production, for example. Non-steel fermenters, do you also use some ceramic fermenters or something like that?
No, I mean at the smallest scale, they're glass fermenters, for various reasons, particularly around cost. But at the largest scale, they're all stainless steel fermenters.
When do you speak of full capacity utilization of fermenters because you have plant shutdowns, for example? These are certainly multi-purpose units, so you have to purify them, etc. When one does speak about full capacity utilization, it's 50%, 60%, including plant shutdowns for purification.
Yeah, so I mean, to the previous question and answer, we have currently one of our 10 meter cube that's near on fully occupied. Mm-hmm. The second, we're getting a lot of interest there, and it is ramping up in terms of the capacity. And in terms of that kind of 18 months to 4 year time, 2 year timeline that I indicated for full capacity, that kind of includes our shutdowns. I mean, we have maintenance programs that are essentially twice a year shutdowns. One of them is a kind of 3-day, another is a week, and all that is included in those kind of capacity indications that I provided.
Thank you.
Lots of information has been given on downstream processing and strain development, but you also have to demonstrate that the mass protein or enzyme that is produced, that this is homogeneous, that there are no heterogeneities. Is test development part of the custom agreement, or do you leave it to external sources or even to your customer? What about in-house test development?
Yeah, that's a really good question. And yeah, absolutely, it wasn't mentioned here, but I suppose that's in kind of the black box behind the scenes capability that we have, which is around quality control testing, and that points to the enzyme activity, the purity of the enzyme, to side activities, also ensuring we hit microbiological specifications around metal content that's particularly of interest for the food industry. And all of that capability resides in-house. In terms of kind of product purity, typically with customer contracts, we have a certificate of conformity and a certificate of analysis that's pre-agreed as part of that contract deal, and all of the specification requirements are mutually agreed within that to ensure ultimately consistency for the end user. Part of the question, I think, or part of the answer is also around the application consistency and ensuring that the enzyme product that we produce performs consistently within the application irrespective of the C of A activities. That's a real strength as well across the BRAIN Biocatalysts area. I mean, the markets in which we operate, you know, as you've heard from Arian, bakery, dairy, beverage processing, another area which is essentially fats and oils. We have lots of application expertise within the business, whereby we can prove the efficacy, the efficiency, and the consistency and reproducibility of the enzymes that we develop. And also operate very closely with customers to make sure that when we say in our application trials it ticks all the boxes, that it does exactly the same for the customer. So application expertise is a key part of BRAIN Biocatalysts, absolutely.
There is a question from the online platform. Is the CRO business really an attractive business with regards to revenue potential and margins? You can't really scale it. The scalable part for me seems to be the BRAIN Biocatalyst product development. How do you prioritize those two? So on the CRO business, what we can do is make our CRO business more efficient, definitely. This is something that we are working on continuously to convert more projects per time. This is something we can do. Of course, the opportunity to generate long-term businesses with regards to licenses, for example, is also very interesting for us to have long-term revenues and not being dependent on the projects, only on the projects. What is really important for us also with regards to the CRO business is that we get insights into actual market trends, new market trends. So we see what the market outside of BRAIN Biocatalysts is looking for, which market trends can be seen, and so on. So besides the revenues, it is also very important for us to get a feeling for the market, where is a need that we can, for example, also fulfill. In general, but this is something, Andy, you can maybe comment on the product for scalability. The products business is probably better suited.
Yes, I'm just trying to merge your answer to this particular question here.
Mm-hmm.
Yeah, I mean, in terms of the CMO business, I think the question here is the CMO business really scalable? And the second part of the same question is the product business seems to be the key part of the business to prioritize, because it essentially clearly is scalable. And the essence of the question is, how do we prioritize? We work very closely together. We are pretty much on the phone daily. We are accountable for ensuring that strategically we deploy our resources to maximize long-term revenue. But we don't do that alone. We have here what we call a new business development team, which, ignoring the description, in essence is around strategic prioritization of resources. And that's composed of Alex, myself, and Arian, and Michelle Snyders, and Martin, who's at the back of the room. We meet quarterly, and it's essentially to do exactly that, to make sure that we are apportioning the right resources in the right direction to please the customers in terms of CO projects that we've either won or want to win, to ensure that we have the right resources to deliver the CMO projects that we're involved in, but equally to retain a ring-fenced amount of resource to drive the products business forward. And for obvious reasons, essentially what Arian said, the products business that's our lifeblood, and continue to be so in the future. That is a key area for prioritization. So thank you, Marcus, online for that question. Hopefully, we've answered that for you.
No more questions at the moment. There's one more.
But this is one maybe more to Adrian about you differentiate your customers with regards to size in a way, and mentioned that you are more focused on the small and medium ones. What about stickiness of the customers in general? I know it from the pharma CDMO business that there are, along with regulatory requirements, you have usually a very good stickiness to the customer, because sometimes the production process is part of the dossier. Is it in your industrial area? Is it the same stickiness, or when you compare your customers with the bigger ones or the even smaller ones, is it the best possible segment?
So I said a couple of things. Is the microphone on? Yeah. I said a couple of things. First, we work with the biggest companies intensively together, but the most revenue comes from the SME. So that just to segment that, which means that we do a lot of business with the large companies, but the core business is with the SME type companies. That's one. With regard to stickiness, you're absolutely right. And the CDMO business is sticky because of those reasons you mentioned. In general, I would say that the stickiness of our customers is really high. Is really, really high. I mean, Darren, you can address it in your talk, but customers typically stick around for years, up to 10, 15 years for sure. In and out, we barely see, it gets a little up, a little down, which also means when you're in a customer and they do well, we do well. If they do poorly, we do poorly. So there's some of those things around. But in general, we are about long-term partnerships. That is really what the business is. In biotech, that is really important, and especially for us, is critical. So those long-term partnerships are critical, which is why on the CRO, CMO relationships and then building that into a product offering is a very long-term offering, essentially, that we're doing. So happy with the stickiness, love to have more of them, and that's what we're building. Darren, you can absolutely further elaborate on that. Is that?
Any more questions?